Okay. Hi, everyone. So I guess there's probably a few more people who are going to trickle in, but I'll kind of kick things off. Yeah, welcome to this Sphere of Living Systems seminar. And we're very lucky today to have Dr. Madhu Venkadesan, who runs the Yale Biomechanics and Control Lab, here to speak to us. So Madhu works on a bunch of really kind of fascinating problems, which range from the biomechanics of feet and fins of fish to things like optimal throwing strategies and traversing rough terrains, and all sorts of interesting problems which kind of have to do with biomechanics. And yeah, so I think today he's going to be talking to us about the work he's done on feet and fins. And so I should say, Madhu has very kindly offered he's happy to kind of have one-to-one chats with anyone who's kind of interested. And so I believe he's put his details somewhere in the talk. And so if you're interested, please do get in touch with him to kind of have further conversations if you want. So if you want to ask questions, I think you can put them into the Q&A, which we'll try and monitor, and we'll probably ask most of them at the end. And if you're happy to be unmuted and want to ask your question live, as it were, then you can just say so in the Q&A or the chat. And with that, I'll hand over to Madhu.
Thank you, Sami, and thanks to you for inviting me to this seminar. A webinar, I should say. Traveling to all the way down under with teaching going on would have been impossible. So I'm delighted that there is this opportunity to present to this group. I've followed various people in your group and things you do over time, and this is really the first time we're getting a chance to interact. Let me dive in and first acknowledge the fact that people made a lot of our work happen, and these are some of the key people in my lab, and I should say used to be in my lab for these two who went on to bigger and better things and got jobs and things of that nature. But not only the people, I wish to also acknowledge various funding agencies which made a lot of things possible in my group. NSF, the US National Science Foundation, from the outside is a big monolith, but inside it has many, many, many complicated hierarchical subdivisions. And among those, there's one program in the engineering side called mechanobiology and biomechanics that funds some of my work. There's a robotics program and a program primarily for biologists, and this is called the Biology Integration Institute. And that's by way of me acknowledging how many different communities have enriched how I think about questions and taught me a lot of things. And then there are funding agencies that I wish to acknowledge, like the Human Frontier Science Program, who in some sense told me that they would like to fund things that nobody else would fund. So I was glad to fall in that bucket and find their support. And various students have been supported by many other funding agencies, including foundations. Before I launch into feet and fins and so on, I want to give you a very brief view of things that happen in my lab. Feet and fins that I'm going to tell you a lot more about today. I will flash this slide because the talk is being recorded and it's virtual. You can find some of the key citations related to our work. That's on the foot. There is a preprint of a paper in review that you're welcome to look at, work on fish fins. But here I want to make a plug for a new institute, or designing a new institute that's funded at Yale. And as part of that, and as part of our ongoing work in the lab, we're looking for PhD students, postdocs. If you're interested in these topics, especially on topics of interplay between mechanics, evolution in biological systems, please do reach out to us. Other than feet and fins, a lot of my lab tends to think about, well, I forgot to play that video. I like strange organisms that do unusual things, and that's a fish that walks pretty well on land. Other than feet and fins, we think about how mechanics affects control, particularly in the biological context. We think about it using very simplified models of, say, running, throwing, We do experiments in the lab using a combination of sensors that can measure energy consumption, high-speed video, but we also conduct experiments on mechanical toys that we build that are inspired by things that happen in biology. I suppose that video is not playing. You'll have to excuse me. That video shows frogs jumping and doing crazy things. The YouTube link will still work if you are ever interested in seeing frogs doing amazing acrobatics. But our thinking from how mechanics affects control naturally went towards thinking about how mechanics of the prime mover of animals, muscle, works. And that topic very naturally went down from whole muscle down into its microscopic structure and asking questions about how bulk behavior is dictated by some of the microscopic mechanics. Of course, this is a topic with at least 150 years of continuous work. And so there's a lot happening, and we are contributing some small pieces to it. And there's a preprint out fairly recently, which you're welcome to look at, on how we think about rheology of muscle and tunable materials in general. Let me now dive into the main topic of today's talk, which is feet and fins. Almost all of this was funded by the Human Frontier Science Program. If you want to reach out to me, if you point your cell phone at that, it should take you to my lab webpage, or you can type that in, where you will find my email address and many other ways of getting in touch. These are some key references related to things I'm going to present today. You're welcome to look for details in there. What I'm going to talk about most of the time today is human feet, and the question of when I push on the foot, on the ground using my foot, using my forefoot typically, why doesn't the foot bend like a thin elastic object, for example, would? What makes the human foot as stiff as it is? And the story of the human foot and debates around it are many times focused on the arched structure of the human foot. And there are two prominent arches that you can see. One is called the longitudinal arch that runs along the length of the foot. The other is called the transverse arch that is transverse to the longitudinal arch. And when I'm pushing with the front of the foot, these arches are thought to contribute heavily to making the human foot as stiff as it is. Roughly 100 years of work has worked out how the longitudinal arch contributes to the stiffness. Our little contribution was to use analogies like this, where I can take a thin sheet of paper. This will be my only prop today. And I can curl the sheet of paper and it becomes really, really stiff, even slightly. And we essentially show that a very similar effect works in the foot. Of course, its manifestation is a little bit more complicated because neither is it isotropic or homogeneous. But the principle is essentially the same as why a thin elastic sheet becomes much stiffer when you slightly curl it. And at the very end of the talk, I will show you how a very similar principle surprisingly works in fish fins too. And surprising because fins and feet, there are some superficial similarities, but they are quite different, and the organisms use them in very different ways. With that, in a sense, I am done with my talk. That's really all I want to tell you. But the rest of my talk, I'm going to try to give you evidence and walk you through how we are thinking about this problem. To better understand why this deep interest in feet, it helps to ask what are humans? And that's where I'm going to start the talk with, where I will tell you a little about how foot arches have been viewed and what are the debates surrounding it, and how we tackle this problem using models of different varieties, biological experiments. Today, I will only present one subset of biological experiments. Ali has done experiments with living human subjects, and in fact, right there, close to where many of you are located, are some of the biggest experts on foot biomechanics who have done amazing experiments with living humans. And I will apply a lot of that to fossil data to see what it tells us about the evolution of the arch. which will really wrap things around and bring us to why did we start thinking about the foot itself. At that point, it'll mostly be obvious to you what I'm going to say about fish fins, but the fish fins have a little bit of a twist. I wish to acknowledge that a lot of this work was done in collaboration with Shreyas Mandre, who is an applied mathematician, primarily doing a lot of PDE theory and fluid mechanics, who's at Warwick, and Mahesh Bandi, an experimental condensed matter physicist. And Carolyn was a postdoc in my lab, and Ali is a soon to graduate PhD student in my group, and various other people who provided materials and intellectual input to the work. So what is a human? Let us start there. And one way of trying to respond to that question is to look at an evolutionary perspective. That's time in millions of years ago, that's today, and around 7 million years ago, we think the lineage that led to chimpanzees and bonobos split from the lineage that led to humans. So we and chimpanzees are alive today, and our last common ancestor was around 7 million years ago. In that subsequent lineage, dotted lines I used to represent debated ancestral relationships. There is not too much debate in terms of the relative timing of these species, but I want to draw your attention to some key species, and which are deliberately color-coded for how biomechanics in the context of evolution here works. People use, say, a chimpanzee as a model for Australopithecus, although that's not exactly the model to use, because Australopithecus was already bipedal and walking bipedally pretty regularly. Chimpanzees, of course, can walk bipedally, but that's not how they spend most of their time on the ground. We also think that Australopithecus could climb and sprint, but a lot of these are a combination of analogy, looking at fragmented anatomical evidence and comparing it with living species, and a very arduous but rigorous process of building up this narrative. But more recently, around 2 million years ago, is when the genus Homo arose, and Homo erectus, a common name examples are Turkana boy or Java man, we think could not only walk, but became quite an expert at endurance running. So humans are incredible at long distances when they have to run. And there are many features that make it possible. One of the key features that makes it possible is we sweat more water than pretty much any other animal on the planet, which lets us manage heat in a sensible way. There are debates, but mostly we think that the bipedal posture helps us be a good runner. And what all of that running, throwing let us do is get more calories. And the story of human evolution in some ways is the story of getting enough calories to support this organ among many other things. But our brain is an expensive organ. To give you a sense of how expensive, I'm not sitting very still. I'm moving around when I'm doing all this. I'm a heat engine. I'm dissipating about 70 watts, and about a third of that is just that organ. Compare that to, say, a squirrel or a chimpanzee. The brain is taking up about 10 to 15% of their sitting heat. What made walking and running and many things possible? There's many of them, but of course, it's the foot is a key part of it, because that's the ultimate interface to the ground. So let's think a little bit more about feet. Let's look at, can I get a confirmation from someone that the video actually is visible? If this is not there, I won't.
Yeah, it is visible to us.
Okay, great. So this is a person walking on a treadmill below, running at a fairly gentle speed of about, I think that's about 3.2 meters per second. And what you see is when we push off with the ground, the foot appears to deform a little bit. The red skeleton is meant to help guide your eye and track how much the foot is deforming, but not very much. It's very hard to pick up on the foot deforming. But to give you an indication of forces in running, when this person is just pushing off with their forefoot, the forefoot's experiencing multiple times their body weight. In walking, things are a lot gentler, but even there, you could end up experiencing a little over one body weight. And in both cases, the foot acts like a relatively rigid thing. Of course, it's not rigid, but it doesn't deflect that much. Okay, so maybe that gives us a hint as to what's going on. And in this view, you can already see that the foot has this arched structure, and that's called the longitudinal arch. And just by correlation, there are people who have very flat feet, and some of them, in fact, many of them face difficulties doing what this person is doing here. And if they try to do that, they will start feeling pain down the middle of their foot that's trying to bend. So we think the arch structure of the foot has something to do with our ability to walk and run in the style that we do. And that in turn, we think has something to do with what makes us such good walkers and runners. So we really want to understand what allows the foot to remain as a good rigid structural member. But there's many debates on this topic. I can't do justice to it in any reasonable amount of time, but let me point you to some examples of the debates. That's just a review paper that's charting many of the controversies, but let me draw your attention to one controversy. How do we know that Australopithecus, for example, was a walker? Well, this beautiful preserved footprints in a trackway, and if you pay close attention, here is an adult that's stepping, and next to it is a juvenile who's stepping. And if you make a more detailed analysis of stepping in clayey soil, this looks as if this animal has a deeper imprint towards the heel and a deeper imprint towards the forefront, but not so much anywhere else. So walking a lot like we do. But pretty much every evidence we have for what type of foot that animal had based on fragmentary fossil evidence is it had a longitudinal arch probably, but fairly weak, if at all. So what's going on? Why is this relatively flat-footed animal able to comfortably walk in the style we do? If you take flat-footed people and ask them to say, lift their heel up, which a healthy person can, here is a diabetic person with a flat foot, they can barely lift their heel up. And if you pay a little more attention to what's happening in the skeleton, you'll see something that's analogous to, this is a bonobo that's trying to walk. And you see the middle of the foot where the white arrow points, there's a sharp bend. So unlike most healthy human feet that don't bend in the middle of the foot, flat feet appear to bend in the middle of the foot. And if you make such a person walk on a pressure mat, you'll pick up high contact pressures around the middle of the foot. But not every flat-footed person, meaning when I look at the longitudinal arch and I say, what's going on? Is this person flat-footed? Will they be able to lift their heel? Not everybody has difficulty. So there's all sorts of conflicting evidence, but a lot of these debates have just surrounded what's called the medial longitudinal arch, what's towards the middle of the foot. But the transverse arch, or more technically, the transverse tarsal arch, these are called the tarsal bones and the arch formed by them, that's really not been studied very much. Not understanding foot in all its complexity, a mechanician such as myself, who can think about simpler things, speculated that maybe the transverse arch has something to do with stiffness, the thing that we've not thought about. Why stiffness particularly, besides the obvious observation that lets you walk in the way we do? I wish to, that was the statement I made earlier, but this is a very nice paper from Takahashi in 2016, where what they did was ask people to walk on a treadmill, and they measured the mechanical power associated with deformation of the foot. So they used something like what you're seeing in this video, dots to track the external surface of the foot and estimate how much mechanical power is going in and you're getting out of the foot. Negative power means you're flattening the foot essentially due to external load. Positive power means you're returning power back to the body, which helps in propulsion. And what they did here was attach insoles into the shoe and kept increasing the bending stiffness of the foot. And what they see is as the foot gets stiffer, it deforms lesser, not surprisingly, but also helps you recover more of the work you put into the foot. That story has a lot more subtlety, more higher stiffness is not always better from metabolic energy consumption, it looks like, but to some degree, sufficiently stiff foot is apparently needed to do this. And remember, energetics is a very big part of what lets us be human. How do people then go about studying this? One of the main findings that has driven our thinking about the longitudinal arch, conducted experiments with cadaveric feet. It's a really clever experiment, because how do you take a cadaveric foot and simulate this kind of loading? Well, what's happening when you walk and run? The ground's pushing on the front of the foot. The Achilles tendon is pulling up on the heel when I'm just pushing off. So in this setup, they took an Instron material testing machine essentially and stuck a truncated cadaveric foot in it. And there was a heel contact, which is mimicking the line of action of the Achilles tendon pulling on it. And this contact is mimicking the ground contact. And they conducted load displacement measurements. The blue curve is an intact, in the sense the foot is intact. None of the structures inside have been disturbed. The red curve is when this thick band of tissue called the plantar fascia were cut away. And the plantar fascia behaves together with the skeleton of the foot like a bow and a string. The bow intrinsically has some stiffness. Removing the string causes quite a big drop in stiffness. Just dimensional arguments would tell you that the plantar fascia's extensional stiffness manifests as a bending stiffness of the foot in proportion to the square of the height of this arch. So having an arch lets you amplify the effect of having a stiff plantar fascia. And in fact, when they cut the plantar fascia, the load displacement curve becomes softer, and softer by around 25%. Then they started cutting lots of other tissue inside the skeleton itself, which disturbs the bow, and no surprise, they have major contributions. This was a key way of understanding what is happening and how the anatomical elements of the foot translate into stiffness due to the medial longitudinal arch. When a similar experiment was done with monkeys that have relatively flat feet, don't have any pronounced longitudinal arch, cutting the plantar fascia essentially had no effect. So it's all very consistent with the idea that the longitudinal arch is important. And I'm going to tell you that the longitudinal arch is a big part of the stiffness of the foot, but it's not all of it. In fact, I will show you that the transverse arch plays as much, probably maybe a bigger role than the longitudinal arch. How do we know this? Well, let's take that curled paper experiment and do this a little bit more systematically. So first, we try some numerics. Okay, so the numerics are taking an elastic shell, a continuum elastic shell that's homogeneous, isotropic, and subjecting it to an edge load clamped at the other end. So think of the clamp as the heel and the edge load as the forefoot load. And we measure stiffness as a function of the transverse curvature. But we also vary what the thickness of the shell itself is. These circular dots are the result of numerics. And what you see is broadly increasing the curvature, meaning making it more arched, increases the stiffness. That's the broad trend. The diamonds and the stars are experimental data. Is essentially mimicking that numerical computation using a knife edge load on one end, a clamp on the other, and subjecting it to careful stiffness measurements using a PDMS shell. It's a fat shell, and the exact dimensions, a few dimensions were incorporated in this test. And again, what you see broadly, more curvature, more the stiffness, but there's a lot of scatter in the data. That's a logarithmic scale. On a logarithmic scale, that's a really lot of scatter in the data. So clearly, just curvature is not telling me what is stiffness, because variations in thickness apparently also matter. That's the continuum version. We can also do something that's a little more foot-like. That's inspired by the continuum version, which is, think of, okay, I'm going to go back. I normally have a foot in my hand, a foot skeleton in my hand, but we'll have to do without the skeleton or images of the skeleton. These long bones are called the metatarsals, and they are arranged in a transverse arch because of the tarsal arch. And that's what I'm going to mimic in this setup. The metatarsals are arranged in a transverse arch, and they're connected by springs here, and they're connected by springs there. So that's these springs give it some intrinsic structural stiffness. And what curvature does here is varies the primary bending axis of each of the metatarsals. As I make the foot more and more curved, the bending axis is going to systematically change. And what you see again is the same broad trend: more curvature, more the stiffness. But we would like to understand what is this scatter due to? Can we derive, and it's not a surprise that stiffness is not a function of just curvature, because they're first of all in completely different units, and we would like to have a dimensionless way of thinking about it. And the dimensionless way to think about it is we would like to derive a dimensionless stiffness variable and a dimensionless curvature variable such that the dimensionless stiffness is purely a function of my dimensionless curvature. I will not go into the analysis in full detail, but I'll present a skeleton of it. And the result of the skeleton is the dimensionless stiffness is the stiffness of an arch object divided by the stiffness of an object that's identical in all other regards other than being transversely flat. And that normalized stiffness depends on a normalized curvature, which is a function of the curvature, the length that's being loaded, and the thickness of the shell, or the distance between that spring and the hinge axis. And where this whole effect comes about is because of how transverse curvature affects stiffness. I mean, to be clear, we're talking about really old results. We are talking about results that date back to Gauss. This is Gauss's theorem egregium, which lets you handle a pizza, stick a currency note into a vending machine, or use measuring tape. All of them have the same principle, which is when I have transverse curvature and I try to bend out of plane, I will also have to stretch in plane. So that having transverse curvature couples bending with stretching. And it's that coupling that gives rise to the increased stiffness, but also an analysis of that gives rise to these dimensionless variables. So that's my setup. For now, ignore the right half and pay attention to this figure alone. What this figure is showing is a heat map of what happens to the vertical displacement throughout the shell if I displace the far end of a cantilevered shell by some amount. What you see is a lot of the displacement is really concentrated towards the tip, and not too much is happening in the rest of the shell. So there appears to be a boundary layer, for want of name, where a lot of the action is happening. You can also see this boundary layer in another way. The curved shell is not simply a beam with a greater second moment of the cross section, the I-beam, so to speak, because of course the fact that I have curvature, the neutral plane is further away, and that gives me greater rigidity. If I plot one of the stress components, this is my x-axis, and I'm plotting sigma xx. Sigma xx should be 0 along the neutral plane if it was a linear theory that applies, but close to the boundary, what you see is a systematic departure of the zero stress contour line from just a standard linear beam theory. So something interesting goes on there, and that interesting thing is bending gets coupled to stretching. So bending, you can think about an energy associated with it, and stretching, you can think about an energy associated with it. There's the bending rigidity times the curvature squared times the area, because that term just gives you the energy density. The stretching energy, similarly, it doesn't depend on the bending rigidity, but just the Young's modulus times the displacement square. But now this stretching amount is because of my curvature, multiplied by the total volume in this case, because it's a strain energy density I have. The size of that boundary layer is going to be one that minimizes the sum, meaning the two terms are balanced. And that gives me a length scale, which is square root of the thickness divided by the curvature. And that also gives me a stiffness scale. And I can now compute the ratio of the stiffness to the flat stiffness and compare this length scale of the boundary layer to the total length and derive a dimensionless variable. And that variable is my dimensionless curvature. So now we've done some very simple-minded, but it turns out an accurate scaling analysis. Let's go look at what is the value of this. The value of this theory is evident when you take these data and plot them on a normalized stiffness and normalized curvature scale. What you see is a collapse of all of the data other than these stars. The stars are experimental data of shells that were flat in the transverse direction but had varying longitudinal curvature. And what this is showing is that increasing longitudinal curvature by itself intrinsically is not doing anything for you. Not at all a surprise because of how I'm loading it and how I've clamped it, but it's a sanity check. The longitudinal arch, the effect of why it induces stiffness in a structure is because of the plantar fascia, which there is no analog in this toy model. With the discrete one as well, it turns out the theory is fairly robust in terms of collapsing the data, but you should be wary. The theory is this scaling analysis, and therefore the theory is all for shallow, thin shells. So I want the curvature to be slight, but really shallow, and I need the thickness to be much smaller than all the other dimensions. And of course, as you saw, the experimental shells were neither shallow nor thin in the strict sense of observing it, but clearly the theory is robust enough to collapse those data and also heuristically apply to a discrete version quite effective. All right, so armed with all this, let's ask, can we now, maybe we understand the mechanism of why transverse curvature induces stiffness. Let's see if we can convert this to a test of the theory in feet. For that, I'm going to show you a very brief video made by Ali, which I think does a much better job than me describing it and waving my hand. I will give some commentary. So that's a 2 metatarsal foot that's flat. So the bending axes of both the metatarsals are parallel. When there is a transverse arch and the bending axes become misaligned, clearly the structure is stiff and lifts the weight up. However, the stiffness is because of that elastic sheet that's connecting the 2 metatarsals. Because what happened due to transverse curvature was bending and stretching become coupled, and when Ali sliced through that elastic sheet, these 2 metatarsals are allowed to act independently again. This is the motivation for how I can test the theory in real feet. Real feet, I can't go around finding another foot that's identical in all regards other than the transverse curvature. There's no hope of doing this sort of testing. Or if I start trying to shape the bones, the arrangement's not as simple. And the consequence of all of that is I can't shape the bones without messing with all other aspects of the foot. So we first did a little sanity check where I can take these mechanical models and do 2 things. Either cut the springs in between them, that's called T minus, it's the knockout of the transverse spring. Or I can just take a mimic that's flat, meaning all the bending axes are aligned, and measure the stiffness of both of these for many different lengths and widths and different stiffnesses of the hinge itself over there. And what you see is, well, they're fairly predictive of each other. Just a sanity check, right? So cutting the transverse spring is equivalent to flattening the transverse arch, and that gives us a way of how we can make this measurement and test this idea in real feet. To remind you, the longitudinal arch and the plantar fascia's role was tested by cutting the plantar fascia and made the foot about 25% softer in human feet. In flat monkey feet, the same thing, yeah, about 4%, but it's very hard to even perceive that. We repeated the same experiment. The things that are new in our experiment is we don't cut the plantar fascia. Instead, we cut the elastic tissue in between the metatarsals, which would have otherwise resisted the in-plane stretching. And yeah, of course, the other obvious thing that's nicer about our setup, notice I say nicer, is we can make these 3D pictures. They had beautiful hand-drawn drawings. Okay, jokes aside, we can cut through the tissue that's preventing the transverse stretching, and then ask, well, how is the stiffness decreasing? So it's an analog of the mechanical experiment I showed you. Here is one sample trial. The black is the intact foot. The experimental setup is virtually identical to this. There are a few subtle differences, which I'm happy to discuss later if someone's interested. And cutting the transverse arch led to a drop in stiffness. These are 2 feet that I'm reporting here of around 40 to 50%. I've also reported something that I've called the normalized curvature. I haven't yet told you how I'm measuring the normalized curvature of the transverse arch in human feet, but the normalized curvature in these 2 feet, for example, are around 15. Okay. So maybe there's consistent normalized curvature and consistent contribution of the transverse arch. Let's use this understanding to start asking how is the normalized curvature across many different human feet. These are arduous experiments, and doing large numbers and finding large anatomical variation is quite an undertaking, which we have not yet done, and I do not know who else has done yet. How do I measure curvature in feet? Well, one obvious thing is that I can look at the external surface of the tarsal bones, or really this end of the metatarsals, and estimate mean curvature over the entire curve. The mean curvature going from that end to this end is simply the angle that I accumulate when I draw the normal to that. But there is a nicer thing that you can do, and this is helpful when we deal with fossils, because fossils are often incomplete. When you look at the feet, the distal end rests on the ground, and the middle of the foot has this arch. And there are clear anatomical facets associated with the bone for what's on the ground and what forms the top surface in human feet, and in a lot of other vertebrate feet as well. What that lets me see is, as I go from the big toe to the little toe, the metatarsal is accumulating a twisted torsional geometry about its long axis. For a very long time, anthropologists have known this as a signature of the presence of a transverse arch. What we did was put a number to it. We used the twist angle of the metatarsal arch divided by this width as an approximate mean curvature of the transverse tarsal arch. I think this is about 12 or 14 samples, both live human and cadaver. I think this is 6 here live human CT scans and 6 cadavers, where we measured using imaging this torsion and estimated curvature due to torsion, which is shown here, and also estimated curvature using the external surface, and they are fairly well correlated. So it lets me use the torsion as a measure of transverse curvature. And we now applied this to fossil feet. The fossil feet that we considered, I should again orient you. This plot, this picture has time on this axis. This is today. That's around 7 million years ago. That is 7 million years ago, around when chimpanzee. That's a photograph of a plastic chimpanzee foot. Chimpanzees split from humans. But the period I'm going to look at is between today and around 4 million years ago, when there are a few key fossils. 4 of them have been identified as genus Homo. Naledi, the dating of it, we think is around 200 to 300,000 years. The exact date is to be determined. For reasons of the species appears to have conducted ritual burial, which makes it hard to date these things. Homo erectus and Homo habilis, which are these 2 feet, are dated around 1.8 to 2 million years ago. Afarensis, Lucy, Australopithecus afarensis, what we found is 1 bone, but thankfully that bone appears to be the 4th metatarsal, which lets me apply my curvature estimate. And this is a very enigmatic foot. It's around the same time as Lucy, but due to other fragmented pieces, we know that it is not Australopithecus afarensis, or we are fairly confident it's not Australopithecus afarensis. So then it is yet to be named species. It's named after the region where this foot was found. On this plot, I'm showing you the normalized curvature on a logarithmic axis. That's the gorilla, that's a chimpanzee. These are the two flat-footed monkeys that were studied by Bennett and Alexander in that cadaveric study I showed you. That's humans. The dot is the mean, and I'm showing the middle 50th percentile of various estimates. For all of these fossils, I really have only one sample, but I have to fill in gaps because I only have a partial system. And to fill in those gaps, I conduct a Monte Carlo. And so using a bootstrap method, we generate estimates of expected variation in the curvature parameter, the normalized curvature. Gorilla is distinctly more arched in the transverse direction than all the other monkeys. On this scale, you don't even see where the mean is. So they're very flat-footed. Humans are clearly away from gorillas. Very satisfyingly, the genus Homo are all very close to humans. Two surprises. Afarensis, very different from gorillas, but also very different from Homo. So it's not as arched as humans, but it's clearly more arched than pretty much all the other primates we considered. Bertelli is a bigger surprise because it appears based on this arrangement shown here, which is based on an analysis of the curvature of these facets, has a toe that's sticking sideways, a lot more like our other primates, but its transverse arch appears to be within typical human variation. So we think that the transverse arch contributes quite a bit to the midfoot stiffness, or at least I've shown you evidence for that, but we think that such a feature evolved at least 1.5 million years before the current day Homo appeared. One of the consequences of what we find is we really need, I think, to re-examine even the definition of what is a flat foot. Our current definitions are focused entirely on the longitudinal arch, and there is one other dimension to consider, and maybe even implications for how these things are treated. I think I have about 4 minutes. Am I correct? Or I'm also happy to stop here if there are lots of questions.
So I think you have about 4 minutes left. I think you're good to.
Okay.
Oh,
fish fins. So the same idea we think applies to fish fins. I haven't yet told you why on earth am I talking about fins in the same talk as feet, but I gave it away a little bit with the mud skipper. But the idea there is you're not walking on ground with fins, but you're stirring fluid. This very sophisticated demonstration made out of paper coffee cups, duct taped, paper sheets cut and stuck to it, is a simple demonstration of that idea. The cup below has flat fins that are very thin and very flexible. The cup above has a slightly transversely arched fin. And what you see is blowing air on one half of the cup causes that to stir a lot, which is a way of me saying, if you have stiffer fins, you can stir the fluid better, which is important for fish as they conduct various maneuvers with, for example, these fins, which are the pectoral fins. These are my arm equivalent, or my hand equivalent. And we think curvature-induced stiffness is a phenomenon there. But here, what I'm going to show you today is entirely theory. And we thought about modeling composite structures made of bones, like the rays in the fin, and membranes, like the skin fold in the fin. And at the simplest, like that video Ali made, you can think of a two-ray unit cell. So two rays, their primary bending axes are slightly misaligned, that's curvature, and a thin elastic membrane that connects them. For this two-ray unit cell, naturally, if I try to load them, they will bend outward along the soft bending axis, which will cause the ray to stretch. The ray doesn't want to stretch very much, so it will try to make these rays, the membrane doesn't want to stretch very much, so it will force these rays to bend together a little bit more, which will engage the stiff direction of bending of this. And that competition between the membrane and the ray leads to a fairly simple elasticity formulation. And there's one parameter that governs this entire equation. And that parameter has many sub-parameters. Gamma is a measure of the anisotropy of an individual ray. Theta is a measure of how much adjacent rays are misaligned in terms of their soft bending direction. And you also see a competition between the membrane stiffness and the ray bending rigidity. And that together gives rise to a length scale, which is analogous to the length scale we saw in the continuum shell, where all the bending stretching is happening. That's the region where the bending stretching is concentrated. We can, in the context of fins, it made us realize that there is something that could be happening. One is, yeah, sure, I can take a fin with rays that are arranged and curve it transversely, and that would lead to this misalignment of the bending axis, that bending stretching, blah, blah. But all of this could also happen in a flat fin if each ray was systematically misaligned from the adjacent ray. So because it's a composite structure, a flat fin can behave, so functionally mimic the effect of curvature. So we took a mackerel fin bought from our friendly fishmonger, put it between two plastic pieces, so flattened it and measured it in a micro CT scanner, and used images of the cross section at 3 different locations to measure the principal bending axes, which are given by the second moment of the cross sectional area. That tensor and the principal directions are found from that tensor. What you see is that the principal bending direction of each ray, as you systematically go from one ray to the other, shows a systematic variation. And that systematic variation, you can ask, well, how does one make sense of what is enough curvature or not? That is made sense of by thinking about how stiffness as a function of that curvature parameter. Remember, there's that one length scale that comes, and the ratio of the length scale becomes a curvature-like parameter. If I have no curvature, the changing curvature slightly does not change stiffness very much. If I'm extremely curved, then, or in other words, one way of thinking of it is the membrane is extremely stiff, the membrane will force my rays to bend together. If I have no membrane at all, the rays will bend however they want. There's an intermediate range where changing curvature slightly or changing membrane stiffness slightly will lead to a very big change in this normalized stiffness. We can work out what is the scale associated with when the transition between these asymptotes is occurring. And that results in a design criterion for, if you want to make a structure, how would you make all of these parameters work such that slight changes in curvature lead to a lot of stiffness? That's the design criterion. From the CT images, we can estimate L by L and 1 plus gamma for our sample. And L by L is around 20, and gamma is around 4, so 1 plus gamma is 5. At this point, it's in the eye of the beholder if whether 20 is much greater than 5 to the 1/3 or not. But it's in the right designed range where the mackerel fin, slight changes in curvature will, we think, lead to a large change in stiffness, yet to be measured, or I have not yet told you. So I'll end here. I showed you about feet, and rayed fish fins appear to manifest the same principle, but within their internal structure, not in the same way as the foot. What I think, in addition to reevaluating what we mean about foot curvature and stiffness, I think it's making us scratch our heads and maybe think about common principles for propulsive appendages across animals. And the motivation to think about that is evolutionary again. We are all fish. We are all fish in the sense, around 380 million years ago, there was a branching of one subgroup of fish called lobe-fin fish, some of which became land vertebrates. And there are still a few of that species, but majority of lobe-fin fish are vertebrates like ourselves. So it's very satisfying that there is a related principle working in both of them. In my group, we are now beginning to make measurements and study select species across the fish side of things, to start asking how did an early ancestor which had fins start to move on land and ultimately gave rise to pretty much every terrestrial vertebrate. Thank you.